And when T cells sit on top of the HLA peptide complex, they have this complex footprint shown in red. And so the white part is the HLA molecule, the yellow part is the peptide, and these are the footprints of two different T cell receptors, both of them recognizing virus peptides, both of them mediating immunity to the peptide. And you can see they're quite different footprints. They're reacting to in a quite different way. And this T cell receptor with its red footprint on this HTLV1 peptide is recognizing that through a very different set of molecular contacts to this one. And so we've struggled to understand why is it that when a transplant takes place, T cells recognize the foreign HLA complex. Why do they do that if they're educated and trained to only recognize our own HLA complexes, to ignore our own HLA peptide complexes and recognize foreign peptides when they enter the HLA cleft? So this has always puzzled medical students, that we should have a T cell system educated to interact with our own HLA molecules when they have a foreign peptide, but not when they have a self-peptide. Why do they get totally confused and suddenly recognize foreign HLA molecules on a graft? And we know that the answer to that is that sometimes the graft can look very, very much like self. And this is an actual footprint of a T cell receptor that's rejecting a graft, and here's the footprint of what it's recognizing on the graft HLA, which is a totally different HLA from this one, which is the self HLA. But you can see that the actual footprint looks very much like this one. So there's a red splotch here, there's a red splotch there, there's a green splotch there, green splotch there, etc. So this is a thing we call molecular mimicry. So that when your graft comes into my body, ironically enough, my T cells react against the graft because we find the bits of your HLA peptide complexes that look most like mine, and we react against them and reject the graft. A really odd arrangement. That molecular mimicry can take place at an absolutely molecular level, where this tiny little part of the peptide here, which is a certain peptide with this sequence on the graft, can be mistaken for a different peptide with a different compound here, but one that looks a bit like this. So the mimicry between the graft tissues and their peptides can involve both the HLA and the peptide. So unraveling this molecular story has been quite important to understanding the molecular basis of transplant rejection. Talk about other ways in which HLA genes can control immunity. There are two HLA genes called B3501 and B3508 that only differ by one amino acid just here. And for all intents and purposes, transplant doctors would say, well, they're pretty much close enough, thank you, we'll do the transplant. But in fact, the way the immune system works is that difference is really quite significant. And those two molecules will actually behave quite differently when they're confronted with a virus in terms of what viral peptides will they show to the immune system. And we've just got some data to show you here. Here's a peptide called zebra, and here's B3501 and B3508. And B3501 is selecting this peptide, and B3501 this peptide, B3508 this peptide. Quite different peptides from the same antigen. And so you've got a situation where there's a short peptide that they both ignore, neither of them recognize it. There's a long yellow peptide which only B3501 recognizes, and then there's an even longer green peptide that only 3508 recognizes. So one amino acid different in this histocompatibility molecule, and the choice of viral peptide used to alert the immune system is very different by this molecule compared to that molecule. So these fine-grained differences can end up being life and death matters when you're in an arm wrestle, an evolutionary arm wrestle with a virus. And we know, for instance, that these differences can make a huge impact on your clearance of HIV, which is dependent on showing HIV peptides of the killer T cells. For the last little bit of the talk, I'm going to talk about a totally different genetic control of immunity. I've talked about how these genes control transplant rejection, it's complicated molecular mimicry. I've talked about how these genes can control selection of virus peptides so that we respond to viruses differently according to the shape of our HLA clefts. Now I'm going to talk about how the HLA genes can control drug hypersensitivity. In HIV, there's a drug called abacavir, which is used to prevent replication of the virus. And about 5% of patients who get this drug have a terrible immunological reaction, very similar to the rejection that occurs following bone marrow transplantation. A rejection episode we call graft versus host disease. And that reaction can be fatal if patients are re-challenged with the drug after withdrawal from the initial reaction. Turns out that the 5% of patients who get this have all got this molecule called B5701. They all have an allele, a genetic variant of their HLA-B molecules called 5701, which has a particular shaped cleft and is a unique molecule. So again, immune response gene being linked to an immune response. But how does it work? And the drug is small and not nowhere near as big as a peptide, so it's not behaving like a peptide. It's too small. So we did some experiments where we showed that T cells can react to this drug when it's added to cells that have the B5701. And here they are reacting in this assay where we measure the amount of interferon gamma inside the cells in response to B5701. Now, if you take a molecule called B5703, which has only got these 2 amino acid differences between B5701, there's absolutely no response at all. And if you now just make it so that there's only one difference, you see some of the response coming back, but it's only 10%, not 28%. So this one amino acid difference is impacting, but not completely abrogating the activation. And now if you just do this amino acid at position 116, you completely lose the drug reaction by the T cells to abacavir. So this tells us that whatever the drug is doing involves the cleft, because if you change the cleft, you change the drug reactivity of the T cells. And we went on to show that the drug is sitting in the cleft. The drug actually sits in the cleft. It's this green thing here. And in doing so, occupies space in the cleft, which means that a new set of self-peptides starts to bind the cleft, and the old set of self-peptides no longer binds. And so the drug is altering your immunological signature. It's getting into the cleft, changing the self-peptide repertoire that is your identity to the immune system. It's just changing your signature. It's a kind of identity theft by the drug. And in doing so, you start to look like a gigantic foreign organ, and your T cells start rejecting you. And that is the basis of the drug reaction. And this just shows that the proportion of brand new peptides that are presented by this molecule in the presence of the drug is about a quarter of all peptides. And these figures show that we can see at a molecular level exactly why this HLA type is linked to the drug reaction and not those closely related HLA types, because that amino acid that that destroyed recognition is here, position 116. By changing that, you increase the bulk in this space, and the green drug can no longer fit. So it's painfully simple geometry that explains this drug reaction, driven by genetic polymorphism in the cleft, which in turn, is what drives genetic control of immunity in distinct individuals. So these HLA molecules dictate this unique patterns of specific T cell immunity, including whether or not grafts are accepted or rejected by virtue of differences in the HLA molecules that confront host T cells. They probably are going to explain a lot of drug reaction. We're seeing an increasing number of drug reactions that are linked to particular HLA types. They're generally small drugs that are ideally suited to fit the cleft and change the self-immunological signature or barcode. And HLA and blood group testing are really the archetypal and original forms of personalised medicine. And like Sharon, my research involves a big team, and I'd like to thank all of them, particularly who work closely with people with Monash and large group at the University of Melbourne. Thank you.